timatangia tō tātou hui. Tēnei te mihi kauatu kia koutou katoa. Aha koa kare e tae e koutou te kite mai I taku kanohi nā te mea kua āhua pakaru taku Zoom link. Kei konei au e mihi atu nei kia koutou katoa. Hoi anō ko riro māku hei whakatūwhera I tō tātou hui. Nā reira, kia taunga Manaaki tanga te mea ngaro ki runga ki tēnā o tātou. Kia māhea te hua mā kehi kia toi te kupu, toi te mana, toi te aroha, toi te reo Māori, kia tūturu ka whakamaua kia tīna. Ui e tā iki e, kia ora tātou.
Morena te whānau, ko Sitlach ko Biasna, awa ko Punjab te whenua. Ko sich te iwi no inya hau ko manpritami toko wingoa. No reira tēnā koutou katoa. Welcome everyone to the third installment of the Wānanga series. If you're joining us for the first time today, this Wānanga series is part of a National Science Challenge project, Te Hoinga I Takatake o Aotearoa. The goal of the project is to gather information and develop a te tiriti guided ethical framework for the generation, storage and sharing of a national reference library for all species in Aotearoa, except humans. I'll just hold for a second while the slides are coming through. Here. Excellent. So Holden and I are the project co-leads for this program of work. And within this Wānanga series, we are aiming to develop and create a safe space for exploring the different issues associated with the development of such a reference library. Primarily, our scope of work is around barcode libraries, so looking at barcode sequences, but in the future, this conversation will also help us understand how we should just generally manage our genetic resources. Sarah, next slide, please. And before we launch into, and I hand over to the facilitators for today's Wānanga, I just wanted to highlight that this project has a working group within it who have been thinking and helping develop some of the kōrero that has led to this Wānanga series. So I just wanted to acknowledge the working group folks, so some of whom might be in the audience today as well. And I guess without further ado, I should hand over to our facilitators for today's Wānanga, and they can give you some details about what it's about. And that's David Medyckyj-Scott and Nick Spencer from Manaaki Whenua Landcare Research.
Morena, everybody. He mihi nui tēnei kia koutou katoa hui mai. Ko David Medyckyj-Scott ahau. I'm head of data management at Manaaki Whenua, a new position that was established about 3 months ago. It's my job this morning to facilitate the first part of the session, and then my colleague Nick Spencer, who's head of digital strategy, will be facilitating the question and answer session. The kaupapa or agenda for today. So this is, as Manpreet mentioned, this is the third of a series of webinars. This one is entitled Database Architecture, Process and Pathways, and it's really about exploring the various aspects relating to the social and technical ecosystem of a DNA reference library, including Maori data governance. Next slide, Sarah. So, as I said, this is a third one. This is probably where the topic hits the road, the rubber hits the road, and that we're going to actually dig into some of the technical requirements and the interplay of those with the social requirements, such as what processes to employ, system governance, the standards to use, and the various roles of parties. On this slide, I tried to pull together some of the considerations that were mentioned around technical and social requirements from the first Wānanga, which was held some weeks ago on end use cases of reference databases. So we're going to probably touch on quite a few of these today. but we're aware that there's quite a lot of topics that need to have some consideration. And we're also aware that we need to actually pull out what the essential ones are and make sure we've given the focus to those. So next slide, Sarah. So what we'd like you to do as we go through the presentations and into the question and answer session at the end is to consider the following questions and to put your thoughts on these into the chat. So what do you see as the social technical challenges in the creation of the DNA library? And what topics will be important that we haven't covered today? So we're making this session slightly different to the previous ones in that we actually do want you to engage and provide feedback and your thoughts as well as posing questions. Just on to a little bit of admin. So if this session is being recorded, I think we should make you aware of that. We have 6 presentations by experts in their fields. One of them is a recording because the speaker couldn't actually make the event at in the end. Each presentation will be about 10 minutes in length. And as I'd mentioned, we'd like you to post questions into the chat panel. And in the background, my colleague Sarah and Manpreet will be trying to collate these for the question and answer session at the end. Any questions we don't get time for or cannot answer, we will respond to afterwards. And as I mentioned, my colleague Nick will be facilitating the question and answer session. As we've got quite a few presentations, let's get going. So the first presentation is by Mick Black and Claire Rye. Mick is a professor biochemistry at the University of Otago, and Claire is a product manager at data services from the New Zealand eScience Infrastructure, or NESI, as most of us know that as it by. And they're going to be talking about building a genomic data repository for Aotearoa. So over to you, Mick and Claire.
Thank you, David. Kia ora tātou, ka whanapaki te manga, ka mate te awa, no wakatū aho, ko Mick Black taku ingoa. Kia ora, I'm Mick. As David said, I'm based at the University of Otago. I am also the bioinformatics lead for Genomics Aotearoa, and recently I've become the co-director as well. So today I'm going to talk very quickly to you about the work we've been doing around building a genomic data repository. I won't go through everyone, but it has involved a combined team of people across Genomics Aotearoa and NESI. Some of those will be familiar to you, and some of those are on the working group for this project. A little bit of background. Within the funding agreement with NB for Genomics Aotearoa, we have a statement which basically defines the genomic data repository, saying that we need to actually build a data repository, including bespoke processes for Māori management of Indigenous data, actively populated across all New Zealand genomics research activities. So we've been working on that over the last 3 years, and we've got a Gen 3 instance running on NESI of this production system. Claire will talk a little bit more about the specifics of that soon. We're focused on genomic data from taonga species, so non-human taonga species. And there's a few definitions on there that have been taken from Te Nohonga Kaitiaki, the guidelines for genomic research on taonga species, which were produced as part of a GA project. So I won't go through those, but those give you kind of an overview of what our focus has been in terms of our thinking around taonga species. As we've gone through developing this repository, which has really been created as part of a need from researchers within New Zealand to store genomic data in a way that honours the kaitiaki of the partners they're working with. So we've tried to balance fair data principles in terms of accessibility with care principles in terms of data governance by Indigenous peoples. So a lot of the protocols associated with how we've set things up and how things run were developed by Medyckyj-Scott, who was our first Vision Mātauranga coordinator within GA. And really the key goal there was to create a system that enabled researchers and kaitiaki to fulfill their responsibilities in terms of data. So researchers making data available, but doing so in a way that was through an access mechanism that was controlled by those who held kaitiaki-tanga over those data sets. And there's it's not quite a quote from Ben, so I'm not sure I've captured his words exactly correctly, but the sentiment is that once our data leave Aotearoa, they lose any and all protections provided by the Treaty of Waitangi. So this was an attempt to create a resource where data could be stored and access could be controlled while still facilitating that access by researchers under specific conditions. So I got two presentations on the go. Very quick screenshot here of the front page of the data repository, number of projects in there at the moment, that sort of thing. And probably I'm going to hand over to Claire now to step through some of the more technical aspects of the project.
Thank you, Mick. Tēnā koutou katoa, ko ingarangi te whakapapa rangi mai, engari ko Ōwāraka te whenua tupu, ki Oriwa e noho ana, ko Claire toku ingoa. Tēnā koutou katoa. Thank you for having me here today. So as mentioned, I'm Claire from Nessie. So just in terms of a little bit more about what's, I guess, behind the scenes in the data repository, we have quite a few species within the repository now, including some eDNA samples. And you can kind of see from the graph of data growth, we've had a few big data sets at the start and a fairly flat line for a little while, and we're now sort of seeing growth of some more projects more recently. We've managed to include biocultural and traditional knowledge labels within the data repository. So this is an initiative started by the Local Context Hub, which sort of allows for Indigenous communities and organisations to give a consistent mechanism for governance and protocols associated with data. So this is, I guess, at a first glance, a quick and easy way to see what protocols are appropriate with a certain data set, and then allows to link out to the Local Context Hub for a lot more information around what that actually means in practice and how you can apply for access. Alongside that, in terms of sort of the technical infrastructure, we have kind of two key user groups, our researchers and our kaitiaki. So from a researcher perspective, they interact with a data portal, a browser, and like many other, I guess, a standard data portal, you can search and you can filter to find data sets of interest. As Mick mentioned, this is based on Gen 3 technology, which is open source software out of the University of Chicago. And this is our, I guess, our metadata store. It does or could be used for data store as well. We don't, because of the needing the granularity around access for data. So we have a separate data store which utilizes Globus for the actual sharing of the data files once there's kaitiaki approval. So researchers can submit their data to OGDR, and this is a supported process because it's relatively manual still. So we help the researchers do that, and also it allows us to check that there's kaitiaki in place, so that in the future, when other researchers want to access that, we have a method of going to get that approval. At this stage, we don't have sort of an integrated solution for the management of that process. It's still relatively manual with sort of Google Forms, but we've looked at using RIMS, which is a resource management system that was built. It's also open source, built out of Alexa in Finland, and we've used it on a related project. So we feel like it's probably a good fit for a next stage of development in that area. And then sort of our kaitiaki interactions can be quite, relatively bespoke in terms of how they want to run the kaitiakitanga of data sets. So often that's done via email, but it can sort of take other forms as well. And I guess the last thing I wanted to mention really for today, and I don't expect you all to memorize the metadata schema here, but just to point out, so we have a bespoke metadata schema for the repository. The sort of the data dictionary from Gen 3 is relatively flexible, so we can change this, and you can see that we've added in, for example, the biocultural and traditional knowledge labels as its own node. And so we're currently undergoing a review to look and update this based on some of our newer data sets that don't quite fit into this model as we'd like. So we want to improve the way we model that. But just to sort of call out here that it's quite a flexible schema if other data were to be included here in the future. And then just to call out a big thank you to everyone who's been involved. And I think hopefully we've maybe kept to time.
That's very good. In fact, I think that's well ahead of time. Thank you very much for that. It's quite interesting to see the importance of metadata. That was one of the things that has come out some of the earlier sessions. So that's very interesting to see that, and also obviously the use of the BC and TK labels. I also quite find it interesting that you're adopting technology from overseas. I think that's a good move because I don't think we really can afford to keep building our own. So thank you very much for that. Now, strangely enough, we're actually moving into metadata for the next one as well. So the next one is a recording because unfortunately John couldn't be here. So John is from the University of California, Berkeley. He's going to talk about the Geome, or the Genomic Observatory's metadata base. His colleague Chris, however, will be joining us hopefully between 11 and 12 to answer any questions. So I'm going to pass over to Sarah, who's going to start the video. Thank you, Sarah.
Hello, my name is John Deck. I'm with the University of California at Berkeley and Biocode LLC. I'm going to talk today just really quickly about the Genomic Observatory's metadata base and metadata architecture behind Geome. And just a bit of background about Geome. If you see the logo in the lower left-hand corner of the screen here, the Maori Biocode project logo, that was a project that began in 2006. And it was an all-taxon biotic inventory of a single tropical island where we had researchers from over 25 different institutions all working together, collecting data in a single database. And importantly, all of the researchers wanted to provision data back to their home institutions. So it was very important for the design of the system to be able to think about data integration between the project database, which was the Biocode database, and collection management systems that people were using. So we started thinking very early about data integration, metadata standards, and data validation for the project. And we came up with a system for people using spreadsheets, because field biologists love to use spreadsheets, and it's still the most common way of entering data. But the issues is that we wanted to provide data validation and data integration and use spreadsheets. How can you use spreadsheets and get data in that format and make it computable and make it work with other systems? So when we think about Geome functions, the main functions first is to provide instant feedback on sample data contained in spreadsheets. We want to comply with global metadata standards, use persistent identifiers to aid in linking to external sources, and enabling users to manage data in distinct project spaces. The relevant bits of the technical architecture, currently it's in a Postgres database that is a relational database, but also NoSQL. Uses JSON to describe both the Geome network and project-specific configurations, and we'll talk about that a little bit more later. It has transaction recording, enables rollbacks to any point in time for each project. It's built on a RESTful framework, and it runs on servers hosted by iDigBio, which is a project funded by the National Science Foundation in the US. So to talk about the user workflow when they interact with Geome, the first thing a user does is they create a project. In a project, they choose particular classes and metadata fields and validation rules for their own project. And they choose from a list of data attributes that are community standard data attributes, such as Darwin Core, and minimum information about any type of sequence, which is the Genomic Standards Consortium standard. From there, users can generate spreadsheet templates, and this is where they enter data. So at that point, the user can be offline. They enter data into their spreadsheet, they could load data into Geome, and Geome gives them feedback about their metadata. Does it comply with the set of rules that they set out from the beginning? In addition, the user can take specimen and event photos associated with their specimen data. They can create FASTA files. So if they're doing barcoding, maybe they have barcodes contained in a FASTA file, or FASTQ files for next generation sequence data as well can be linked there. So we have Geome as this, where it says load data, that's the core bit of Geome that does the validation. There's other features that, such as a query interface and an R package that sits on top of Geome. There's functions for working with tissues and working with 96 well plates, as well as optional linking to external projects. And we'll get into the external project linking on the next slide. So as far as metadata classes and modules, it begins with, at the highest level, events, collecting events. And samples are derived from collecting events. And you can take photos of samples or events. From here, users can optionally submit to the Global Biodiversity Information Facility their metadata. They can also take tissue subsamples or subsample data, which can be tissues, and link the tissues to the barcoding module. Users can then submit to a downstream system such as BOLD, optionally. If they have next-gen sequence data as FASTQ files, there's a submission tool to push data to the sequence read archive, which then has, once it gets there, there's a link to EBI. And we're working on currently environmental DNA handling. And what we've been talking about with some people in the community is using biome format files and amplicon sequence variants within those files to characterize everything that is in a particular eDNA sample. There would be a separate taxonomy classification service. This would likely be outside of Geome, but the purpose of Geome here is to contextualize the sample data and the event data and make sure that this data is linked to the ASV data. So when you create a biome file, all you have to worry about is loading in the appropriate ASVs and then linking in your sample metadata that's already in Geome. This is an example of some collecting event metadata where you can see that there's a year collected, decimal latitude, longitude, locality description, collector lists, and then down here at the bottom you see child entities, which are samples. So in this case you can have multiple samples associated with a single collecting event. And if you click on a particular sample, you can see that there's child entities to the sample. And so here there's a child entity of a tissue that's associated with the sample. And if you click on the tissue, there's a child entity, for instance, of a FASTQ metadata file. And then from there, you can get all of this metadata for the FASTQ file. So as you can see, there's metadata attributes for each of these particular classes. And each of them have a unique identifier, which we call a Biocode Commons identifier, the BCID, and a particular link. So you can link to distinct entities, the FASTQ metadata, tissue metadata, sample metadata, or the event metadata. And I think that's about all we have time for. If you want more information, you can go to the website, geome-db.org. You can also send me an email at jdeck@berkeley.edu. Thank you very much for your time.
Okay, well, obviously I'd like to thank John for that, even if he's not here. We'll pass him our thanks for doing that. As I said, there's an opportunity to ask some questions of his colleague Chris at 11. If you have got any questions for John, could you put them into the chat and maybe tag them with John so that we can forward them on? I mean, it's an interesting presentation from the point of view of the data exchange. So recognizing that you actually do need to move information between different systems, and the integration of the support for R so that researchers can actually access the data. So interoperability important there, and also the use of standards, which is again something that we raised early on in the list of things that needs to be considered. Our next speaker is Natalie Forsdick, who's a researcher in conservation genomics at Manaaki, Whenua. We're slightly stepping out the very technical stuff now and looking more around some of the practices and processes. And Natalie's going to be talking about data management practices. And the title of her presentation is Journeying Towards Best Practice Biodiversity Genomic Data Management. Thank you, Natalie.
Awesome. Thanks, David. I'll just get this shared. And hopefully that's all looking good. Ko Nat Forsdick tōku ingoa kei tamaki makaurau aho, tēnā koutou katoa. I'm a genomics researcher at Manaaki Whenua Landcare Research, funded by Genomics Aotearoa. And as you may have seen from these first few talks, genomic data management is complicated and technical. So alongside Jana Wald at the University of Canterbury, I've been co-leading a project that aims to support biodiversity genomics researchers in embedding good data management practices into their everyday work lives. Our Smart and Kind team includes biodiversity genomics researchers, e-research and libraries staff, and those with broad interests in Maori data sovereignty. So today I'm just going to give a bit of a brief overview of some of what we've been working on here. And first off, I thought we'd speak about what is data management. So this is the processes and practices associated with the documentation and storage of and access to data, and the associated metadata throughout the research life cycle. So this comprises the who, what, when, where, why, and how of your data. Now, the challenge that we're working with is that the volume of biodiversity genomic data is rapidly growing. So over the past 20 or so years, genomic sequencing has had massive, rapid technological advances, and we've gone from working with spreadsheets that might be 100 rows and columns to data sets that are in the hundreds of gigabytes. And similarly, technology means that we're ever more connected and researchers become increasingly collaborative, and there's a growing emphasis on the need for collaborative research. And so data is often shared between institutions and across countries. Due to the nature of institutional setups and the division of subject matters, researchers can be siloed from their data management knowledge holders, such as our e-research teams and library staff. And this means that emerging researchers will often struggle to work out how best to set up their projects, and their group leaders will often be unaware of how these challenges, how they're tackling these challenges on a day-to-day basis. In addition, the nature of the science system currently means that employment precarity often directs researchers to focus on the outputs, including published articles, rather than the day-to-day minutiae of data management. So in total, all of these challenges can lead to an increased risk of data loss and misuse. But the goal is that we want to be working in a fair and care ecosystem. So we have these fair data principles, which largely offer technical guidance around the findability, accessibility, interoperability, and reusability of data. And we have the care data principles that speak to how we can interact with Indigenous data. So acknowledging the goals and challenges set out, our team wanted to understand the questions that individuals across the research space might have around the practicalities of managing genomic data. And so to do this, we developed a series of user experience personas. And these allow us to capture different perspectives across the research ecosystem, including postgraduate students just embarking on their research journeys, postdoctoral researchers who might be dealing with the nitty-gritty of the data, research team leaders that are trying to best support their teams in developing consistent practices that meet the international requirements. And we also include e-research managers who can provide solutions for the data management challenges experienced by these researchers. From this experience and exercise, we identified not just the challenges and questions that these individuals might have on their data management journeys, but also a series of solutions that they can use going forwards. And we've summarized a lot of this work in a web resource, the Biodiversity Genomics Data Management Hub. And here we've described the personas, a lot of their background, and a lot of the questions that they might have along their way. And we've also provided a series of modules which do a kind of deep dive into some of the solutions and some of those challenges as well. And this is what we're really hoping will help out emerging researchers and research teams generally in deciding how best to be managing their data and what tools are available to them that already exist. So just to briefly provide a bit of an overview of some of the solutions that we've come up with along the way. We recognize that data management plans are becoming much more commonly required in research funding applications, and these can be a really amazing tool for thinking about how data will be managed across the research lifecycle. So they can be used for stimulating conversations around data management aspirations with our collaborators, Indigenous research partners, and other stakeholders. So in a data management plan, you're capturing information about the data types that will be generated, formats, standards around these, the roles and responsibilities of the people comprising the research team, how the data will be disseminated downstream, how it will be shared and accessed, and what it will look like in the long term once the project has completed and we might want to box it up and set it away. DMPs can be really useful for helping your e-research and library staff in identifying your needs and identifying infrastructural constraints that may arise on this journey. So just a bit of a deeper dive into what might be included in a DMP. It sort of comes across as broad categories of what, when, where, why, how, and who again. And there's a series of different questions that you might want to be asking along the way to understand how that data will be managed. And this is useful for not just those conversations that I mentioned, it's useful for helping you understand the big picture and the long-term scope of what your data will look like. And of course, it's useful for those conversations with the e-research and library staff. And so we recommend that you connect with your local DARL, your local e-research team members early and often, because they're here to help. They really want to help us maintain and manage our data in the best way possible. But you need to be able to provide them with the context and the understanding of what that might look like for you. And you need to know who's going to be responsible for the data, and they want to know that as well. Not only that, but no matter what your position in the team, you can be your own leader in data management. You can be a data management champion. We encourage people to share their learnings and encourage others often. And remember that data management is a practice and not an event. It's something that we want to be doing, integrating into our daily practices rather than saving up for the end of a project. Lastly, the take home, data management is a journey, and we're all on the path striving towards best practice. And it's a complicated pathway, but we can get there together. And I'd really like to thank all of the people that have provided feedback along the way and all of our funding partners. And thank you.
Thank you, Natalie. That was really good, but I'm bound to say that because I'm head of data management. No, that was really good. Thank you for that. I think Natalie would quite appreciate people going to look at the hub and providing some feedback on it, and to her and her colleagues. I also like the fact I'm a great fan of personas. I think they're a really good way of identifying different characteristics of people's and what their needs are and how they should be supported. And it's nice to see also the mention of library and support staff, because sometimes they get a bit left out and just suddenly discover they've got a role that they weren't aware they had. So thank you for that. It was really good. Can I remind people to put questions into the chat so that we've got a pool of questions at the end. And I'm now going to move on to our next speaker, which is Lindley. Lindley Jessen, who's Principal Scientist and Group Leader of Data Science at Plant and Food Research. She's also leading the cross-CRI working group on Maori data sovereignty, and I think she's going to touch on that in her presentation. Lindley's going to talk about implementing Maori data governance into data management processes and where the CRIs currently are on in that journey. Thank you, Lindley.
You're on mute, Lindley.
Lindley, you're on mute.
Apologies. You
Right,
think you
thank
get
you,
this
Lindley.
right after all this time. Kia ora koutou, koloni toku ingoa, no tamaki makaurau aho. So I'm a Principal Data Scientist at Plant Food Research. And when I was asked by David and Nick to give this presentation, I was a little bit not sure what to say because the CRIs are at a very early journey. And David, you can see my screen okay, is that right?
Yeah, you're good.
Good, okay. So what I thought I'd talk today is about some of the things that we've been doing across the CRIs and then some of the problems that Plant and Food have. Let's not call them problems, let's call them opportunities to actually really start to improve our data management processes. But as I say in the beginning, most of the questions, not a lot of answers, because these are slow conversations. These are trying to get the principles right. We don't want to rush into solutions without having thought about all the issues that we're working through. So there's a cross-CRI working group on Māori data sovereignty, and this has been coming together for about 2 years, I think. And initially it was around if we're going to start thinking about Māori data sovereignty or management of Māori data, or the governance of Māori data, if we're all doing it separately within the CRIs, then we're going to be inefficient, and we're not going to help, and we're going to come across the same shared problems, and we're not going to share our learnings. So if we all come together and share within the CRIs, then this can only be a good thing. Initially, it was said, well, why don't we include universities and others? And it's like, oh, that just feels too scary, because this is, the realm of data at universities is even more overwhelming than the massive data that the CRIs generate. So for the moment, it's a CRI working group, and we're happy to share our learnings. So the working group is sponsored by Te Ara Pūtaiao, and Te Ara Pūtaiao are the Māori GMs of the CRIs, or the Māori strategy general managers. And so we've been reporting into them and getting a lot of feedback and a lot of really good kōrero around the space. And we have Holton Hoya and Chris Koreheke from Te Ara Pūtaiao represented on the working group as well. So we've been working for over a year, but I think it's a little bit longer than that. And one of the things we started to think about was, what are some key principles around data for CRIs? And these key principles are things like, we're good PhD partners with Māori, we manage and use and collect data within ethical frameworks, we collect data for scientific endeavours. So the scope of CRIs and the CRI Act is to conduct research. We collect, manage and use data within legal frameworks, and we collect, manage and use and share data for the benefit of Aotearoa New Zealand. So those are draft principles right now. We're still testing them internally, and we're starting to test them with partners as well, EU partners as well. But we tested these principles at a hui within the CRIs in Lincoln about a month and a half ago, and we got a really good sort of feedback about if we were going to operationalize these principles, what's needed, what are the resources and tools that are needed to enable these sort of principles to be operationalized. So we asked for feedback from that hui about what was the sort of pieces of work that we should be doing. And a lot of the feedback was around common consultation processes with Māori by the CRIs. So how do we come as one? How do we have a relationship economy with Māori so that we're coming and co-developing, as opposed to 7 different CRIs all tuning up saying, have I got an idea for you? And more than 7 CRIs, lots of different, all different researchers from the CRIs as well. So how do we do that? How do we address legacy data? Research data has been collected in New Zealand, well, since Māori we can, but in terms of CRI or DSIR, how do we address the legacy data that we've got? What are the principles and approaches for new data? The common metadata or geotagging data that's needed for every data set. And one of the recommendations is that we should start doing location data for every data set ASAP, because if you don't know where that data came from, building those relationships makes them much more difficult. And then there are places where we need institutional support. So how do we ensure this is rolled out by the CRIs? How do we consult with Māori on the best way to make information discoverable? How do we hold the information with integrity? And so the next big step for this working group is to we're thinking about hui with our iwi partners to make sure that we're co-developing our principles, but aligning with whānau iwi hapu priorities. So it's a big piece of work, and necessarily it has to go quite slowly because every CRI is at a different place, every researcher is at a different place, and the relationships with Māori are at different places as well. So we're doing that. The other thing that has just come out that I'd like to put in a plug in is Te Kāhui Rauranga, and they've put in they've come up with a Māori data governance model, and so that's if you Google that on from the website from Te Kāhui Rauranga, you can come up with that report. That's a really useful model of thinking about how to work with governance with Māori, and that's a really useful link there. So I just put in a plug-in for that. There's been some great thinking put in there. Okay, so at food research, we have some key projects around the taonga data space. So some of the things that we are looking at very seriously is thinking about our metadata requirements. So what's the minimal set of metadata that enables good governance? So at plant and food, location data isn't actually traditionally a metadata field. So how do we ensure that the location data is absolutely collected? And then we have issues with metadata about relationships. Relationships change, people's roles change, knowledge is not encoded. So how do we put that into metadata in a way to enable a continuing relationship? We have projects around a lot of our historical collections. So we have a lot of plant extracts and plant samples, and which have been used over time. More recently, these have been developed in collaboration with Māori, but historically less so. And so how do we go back and do that historic consultation and that historic discovery? And so as part of that, we're not quite in the as good as Manaaki Whenua here, but we are working to incorporate the biocultural notices into our internal data registries, and then find a way to have that transparency so that we can start that external consultation process. One of the ones that is a really interesting one for me is we have one iwi who want data repatriation. And actually, it's not the iwi; it's a group of the land block owners within that iwi, and so they want repatriation of data within their whenua, from their whenua, and that's a really interesting question, because from those particular groups of land blocks, we have got a huge variety of data that's there. We've got genetic data, we've got chemistry, we've got different phenotypes, we have land use, we have management activities, we have insect collections, we have microbiome data. And many of these were part of projects that were in collaboration with other CRIs or other universities, or were subcontracts of that. So we're really trying to work through the principles of data repatriation, because just giving data back to the land block owners can put more of a cost on them than benefit. So if the metadata is not correct, if the data isn't understandable, if there isn't the processes to explore the data, if they don't have the right software, then all of these things can incur a cost on the data rather than a benefit. And so we're really trying to think about, how do we get benefit from data repatriation? So how do we benefit from this, rather than how do we just go through it as a process? So how do we get the benefit, not cost? How do we do this with multiple players and multiple collaborators? So many of these data sets are comparative data sets where samples have been taken from across Aotearoa. And so how do we do repatriation of a small subset of that data and still enable the benefit from that comparative data to happen? So currently we're working on developing, with our partners, we're working on developing dashboards around their data so that they can interactively explore the data and understand the project updates and how things are going. But currently the partner wants us to continue to host the data on our servers, so they don't have the cost of the data hosting, but they can get the benefits of the understanding that can come through the data. So that's something that we're just starting now, but I think that's a really interesting project that we're exploring. So that's all I think I have for you today, but feel free to contact me if you're interested in any of this. And if you want to be part of the CRI working group and help us out, we would love to hear from you. So thank you very much.
Lindley, do you just want to mention how people can participate in the development of those principles if they work in the CRI?
Okay, yeah. If they email me, that's probably a good thing. We're currently at the stage of developing some working groups around those big pieces of work that need to happen. So we're actually wanting people who want to be involved to let us know, so that they want to be part of these working groups. So contact me at Plant and Food, or if you're in the CRI, you're a member of Te Ara Pūtaiao would have my contact details, as well as people who are on that working group. So David, Medyckyj-Scott, Aaron Wilton, Jonathan Kilgore at Scion. Names have escaped me, but feel free to get in touch, and I'm happy to.
Okay. So that's really good. I think the CRIs collectively have got a lot of common issues that they're going to have to resolve, and as Lindley says, coming together to work out some practices and processes that are standard across the CRIs is probably a very good idea. You'll also hear in our final speaker, Jeff, who's going to talk about what Manaaki Whenua is doing in this space. So thank you, Lindley. The next presenter, and he's a new face. We've not met Hamish before, but he came strongly recommended from some colleagues in Australia. So I'd like to introduce Hamish McDonald. He's a postdoctoral research fellow at the University of Queensland, and he's going to be talking about database and genetic resources and considerations for access and benefit sharing. So welcome, Hamish. Thank you for speaking, and over to you.
Yeah, hi everyone. Thanks for having me. Is that coming up? Oh, hang on. How about now?
Yeah, looks good.
Cool. Yeah, hi, my name's Hamish McDonald. I'm at the law school at the University of Queensland, and I also work with the Centre of Excellence for Plant Success here in Australia. And today I'm going to be talking about introducing the idea of access and benefit sharing regimes, talking a little bit about the Nagoya Protocol in particular, and kind of discussing some broad general considerations for databasing that comes out of those ideas. So benefit sharing regimes are a set of different regimes which have developed over the past several decades. And these sort of arose out of a concern that countries which were often provider nations of genetic resources and custodian nations of genetic resources were not being adequately compensated for that work. And so these tend to be based on the idea of national sovereignty and ownership over genetic resources. And they operate through a framework of access and benefit sharing. Access meaning informed consent to access the genetic resources, and benefit sharing meaning some kind of reciprocal benefits flowing back in exchange for the use of those resources. As I said, the kind of most relevant one here is the Nagoya Protocol, which implements some of the benefit sharing obligations in the Convention on Biological Diversity. And it's worth pointing out here another one that's sort of relevant in this area is the treaty, the plant treaty, the treaty on genetic resources for key food crops. And that kind of provides a list of important food crops which are not covered by the Nagoya Protocol, but are instead covered by a separate multilateral system. So here, the countries in blue are those which have signed and ratified the Nagoya Protocol, meaning they've implemented it in their legal systems. You can see that New Zealand hasn't signed or ratified, and Australia and the other countries in green have signed but haven't actually implemented the protocol yet. Despite that, there's a number of reasons to still be concerned with the Nagoya Protocol and to kind of consider it when you're working with databases, and particularly with plant collections. The main reason here is that non-state institutions, particularly herbaria, but increasingly things like universities, journals, and funding bodies, are kind of looking more at this data governance and at whether Nagoya compliance has been achieved in data. Ratifying countries are also under an obligation to ensure that anything they import is Nagoya compliant. And finally, it can provide a sort of model for voluntary benefit sharing systems and for kind of setting best practice proactively. So there are good reasons even in countries like Australia and New Zealand to consider benefit sharing and to consider the Nagoya Protocol. But because this is an international framework, which works between states, there are some difficulties with doing this in countries like Australia and New Zealand. And the main one being the need to kind of, how do you conceptualize a resource provider in that instance? Do you think of that as a state or government body, or do you think of that as, for example, a local community or an indigenous community? There's also considerable ambiguity around the idea of, or the phrase genetic resources itself. Most importantly, it's still being debated at the global level whether digital sequence information constitutes genetic resources, or whether this only applies to physical plants and plant parts, which is kind of it certainly does apply to physical plants and plant parts. So the kind of relevance of this sort of thing to something like a DNA reference library clearly does depend on whether or not sequence data and other kinds of related information are considered genetic resources. But even if it only is considered to cover the collection of physical plants, there are still some kind of metadata, access restrictions, and data governance considerations which might come into play when you're working with or designing databases. So in terms of access, this was sort of talked about in some of the earlier technical presentations. Whether or not access is covered by access and benefit sharing regimes legalistically depends on whether or not genetic information and sequence information falls within genetic resources. But in which case, if it does, access and benefit sharing will kind of mean that there are quite strict rules on who is able to access this kind of information from databases and online systems. But as was talked about earlier, there's good reasons for having those kind of access considerations in place anyway. And I don't think I have much to generalize on this, because it obviously depends very much on what sort of database is being developed, who's involved in it, and who would be appropriate to give access to. In terms of benefit sharing, there's a number of kind of different metadata fields, which have also been mentioned in previous talks, which can be collected to ensure that sufficient information is available that access and benefit sharing can take place at a later point in time, and also to link back to any collection that has occurred and to ensure that the right information has been stored to enable that at the point of collection. And it's important to consider both, like, whether or not these fields should be included and how they should be implemented, whether or not these should be mandatory or not. The most important information to collect, as I think Lindley noted, is the location and the date of collection, as specific as possible for both of those, and also any permits obtained. It's important to collect information about things like biodiscovery permits and also things like environmental permits, because these are sometimes can be evidence of benefit sharing with the state. And they're also relevant to access in that they are kind of a form of permission from the state for that collection. Other possible fields that might make sense depending on the kind of database involved are details on the land ownership at the time of collection, any agreed benefit sharing that's been concluded, and any kind of associated traditional knowledge with those genetic resources. And I just wanted to flag here the biocultural and traditional knowledge labels, which are kind of one way of implementing some of this custodianship information, and one way of sort of tracing this as it moves through different data systems. Finally, the care principles were also discussed earlier. So I won't go into these, but I just wanted to add maybe that these can be a valuable framework for assessing data context, data governance in broader context as well. And they're things that probably should be considered for any kind of database or data system. I think I've run a little bit under time, but takeaways are that even where Nagoya hasn't been ratified, or where there are no legalistic access and benefit sharing regimes in place, it's still worth considering that at the point of designing and using databases. How much this actually governs databases in a legal sense will depend on how genetic resources are eventually defined internationally, which is probably a process that will take some time and is unclear which direction that will go in. In the meantime, the inclusion of plant collection metadata about the location of collection, the time of collection, any kind of permits and agreements surrounding that collection is particularly important to collect for facilitating future access and benefit sharing. And I think that's something we should be thinking about now. Thank you very much.
Thank you, Hamish. Very good. I guess one of the takeaways from that is the sort of legal complexity and the different views there are around this topic. And as you pointed out, particularly considering the local context, which in New Zealand is very important because of our relationship with Maori. The other thing I noted was, yet again, we get the mention of metadata, which generally scientists are pretty notoriously poor at doing, certainly in the areas that I come from. And so the idea that we require them to capture even more provenance information is really important. And I think one of the things we have to consider going forward is how do we make that easy? Because it's not something that our scientists like doing. So thank you very much for that. That links nicely into our last presentation, which is been given by Jeff Ridley. Jeff is portfolio leader for biota at Manaaki Whenua. And he's going to talk about what Manaaki Whenua, particularly around our biological collections, has been doing with respect to thinking about Māori concerns and the relationship to treaty and DNA. So over to you, Jeff.
Thank you. I'll just try and find my screen now. Cool. Can you see that?
Yep.
Cool. Hey, thank you. I'm Jeff Ridley. I'm portfolio leader for biota at Manaaki Whenua. And I want to talk today about how we're interacting with some of the stuff through the T3D with our nationally significant collections and DNA. So Manaaki Whenua in 2021 made a commitment to treaty. about the principles of the Treaty of Waitangi as defined by the courts and the Waitangi Tribunal and reaffirmed by the Arawhiti and the Cabinet Office Guidelines of October 2019. So we've made this real commitment, and in particular, we've made a commitment around the biological collections, and this is be a place where we're going to play with, to implement, and to see how we're going to make all of this work. As I say, sitting around the collections. Now, we, on the right-hand side there is our opening page to our collections. And in thinking about the collections, we've developed a set of principles when we consider the treaty and what we're supposed to be doing. So right up the front there, we've made that committed to uphold the treaty. The collections are there for the benefit of Aotearoa. We need to be strategic and responsive. Our collections need to be curated, reliable and secure. They need to be accessible. There's no point having collections if they're not available to people to use. We need to be collaborative and well connected. So all of the stuff that we have, we need to make sure it's that usability, being connected, making it available for people to use. And it needs to be underpinned by critical capability, because the basis of nationally significant collections is they're a reasonably authoritative place to go. They'll never be perfect because they're working collections, so things will change, but we need to have that capability sitting under them to be able to show that they are valuable to people. So we developed those principles over the last couple of years since we've made that commitment to the treaty. The collections themselves consist of physical objects and living things. We have 5 nationally significant collections. We have the plant collection down in Christchurch. We have a fungal collection in Auckland, and we have the insect collection in Auckland too, which consists of dead material, essentially, of herbarium collections, of fungarium collections, of the pinned insect collections. So, and in size-wise, the insect collection is up around 6 million specimens. The plant collection around 110,000, I think it is, something like that. So they're quite huge collections of material that we've got. And that's the objects, the dead material. We also have living collections. So we have the Harakiki collection down in Lincoln, maintaining what are basically land races of important land races to Maori. We also have a significant culture collection of bacteria and fungi, which we maintain under liquid nitrogen, which is around about 22,000 isolates, I think. So a significant collection, a significant component of New Zealand's biota in these collections. Looking at the collections themselves, for instance, when we think about the data that rides with the collections, the associated data. So we have that what was mentioned somewhere at the collection event. So in this case, looking at the insect collection, we have this pinned insect here. And this physically, historically, the collections were all started in the early 1920s. So they're all reaching a point of celebrating 100 years. And so we have, and that accounts for the size of them as well. So we have a pinned insect here. Now, traditionally, an insect like that carries its data with it. So what you see next to it is over the history of that collection, the data that's been recorded and pinned with the insect. So if you look at the left-hand side of each one of those little labels, you'll see a hole where it was on the pin that the insect was on. So that's the kind of data that might sit with an insect. If we look at plants, the amount of data increases rapidly. It can be much more substantial because we have, in this case, a dried specimen, pressed specimen glued to a sheet, and over the history of that specimen, you can see various labels, various pieces of information have been attached to the sheet. Now, obviously, with time, we are now trying to capture all of that physical data that's written on the sheets and the specimens and get it databased. So again, as I say, different kinds of information there. I think the last speaker talked about the importance of provenance, where it was collected, when it's collected. All of that sits there on the sheet. It's a fundamental part of collecting specimens is that material is, that information is recorded. If we look at, say, the fungal collection, again, the same kind of thing. Up there, the top left, we've got the actual physical sample as it exists in the collection. With that collection, we'll write photographs of it in its fresh state. There might be microscopic photographs of the tissues, etc. There's the data that was collected in the field, and there might even be additional material in the form of glass slides and other physical material that was derived from the specimens. With time, again, as we start to think about the physical collection, those collections generate information, other data. And so on the left-hand side there, we've got the field collection, the physical specimen that we keep. Around it, as I've just shown in some of those previous slides, there may be digital images of the specimen at various levels, macroscopic through to microscopic. And there'll be the information about where the specimen is, where it's recorded, its number, so we can relocate it, and things like that. So that's we call the primary extension to the extended specimen concept. So that's the basic information that will ride with the collection. With time, we may collect other information around morphological data, ecological data, etc. But the one of obviously interested in this meeting is that molecular data, the DNA, the sequence data, the barcoding. And so again, that is something we collect, and somehow we've got to make sure we start to integrate it within the system, keeping all of this information tied back to the physical specimen. And the importance of a specimen is I pull this quote out of somewhere: a single plant specimen mounted on a herbarium sheet may be analysed in multitude of ways to yield data on flower morphology, DNA for applications from systematic studies to genome sequencing, and isotopes for analysis of nitrogen to understand the mechanism of phenology in relationship to nitrogen uptake. All of our specimens have that capability. So the depositing of a specimen is a marker in space and time for a species, and therefore all the data that sits around it has the potential to be used. And but one of the problems is around when we start to think about the molecular data is that I came across this study. I was looking to see how good the existing data systems are, etc. And when this particular paper that was published last year, I think it was looking at insects and trying to do those insect identifications against Bold and GeneBank, and finding that there was only a 35 to 53% accuracy. Now, one of the things about a collection is that when you create the barcoding information, the sequence information, you can always go back to your specimen and check it to make sure you've got it right. Or if your identification was wrong, if sequence data and barcoding data is lodged and there is no specimen and they got the identification wrong, that information becomes so much junk and actually gets in the way of being able to do what you need to do. So specimens become really important in defining what should be going into the library, so that you've always got an opportunity to go back and validate that material. In our own system, again, we don't. We're grappling with how we're going to deal with it, the bulk of information that we are producing, and here's one example of trying to make data available, but not necessarily at this point making it searchable all of the time. And here we have a one that's come out of iNaturalist. We've got the photos and the locations up there. It's been taken back. We've taken good photos of it, so we know what the thing was that we looked at. We've put up the DNA barcode, and if you look at the bottom right-hand part of the screen, you'll see that there's a PDD voucher number, so we can link all of that back to the collections and be able to validate it all. The other thing we're doing in the collection is with bicultural labels, again, to make sure that we're moving towards our compliancy and being good partners with Te Tiriti, and taking into account the fair and care goals, as well as the NZ goals around data management. We've been working with trialing bicultural labels as our first step in doing this. And I think somebody's already defined bicultural labels are a means for Indigenous communities to identify and maintain provenance, origin and authority over an object and data generated from their lands and water. So this is something that we're really keen to be able to do. So if you look at it, you pull up one of our specimens, if you went into the systematics collection database that we have online, pull up a specimen, you can see what it looks like. There's the data that we've recorded in the database. It will relate back to all of those labels you can see on the specimen. And on the right-hand side of the picture of the database screen, you can see we've got the bicultural labels that have been applied to this particular specimen. About cultural labels, in this case a provenance one, you can see what's been included. And so these have been applied to some. And where we're at the moment is we've been working with 3 iwi. And so we've got, say, with this particular one at the top there, with the iwi has applied those particular labels to around 6,000 specimens of a, of various kinds of labels that they see appropriate. We've also applied bicultural label notice to all of the collections that we have up on the database. So it's 675,000 objects now have a bicultural label on them, telling whoever looks at them that other labels could be pending, other notices could be pending, sorry, and to be aware of that when they're thinking about what they're going to do with the data. And so if there's DNA data, that will also, the labels will apply to that as well. And so I'm going to finally wind up when we're thinking about a DNA reference library. DNA must not lose its digital connection with the objects and specimens it was derived from. That's really, really important from our point of view, I think. What else is there, I think? Yeah, as I said, I think DNA without associated objects is a little more than junk sometimes. So again, when you're dealing with the range, the huge number of species that we're dealing with, it's, again, can't overemphasize the need for that connections. With smaller subsets, we're only dealing with maybe a dozen, 20, 30 species. It's fine not to have specimens sometimes, but certainly with us, when we're trying to deal with the whole biota, we need to be able to pin back to those collections. And the other thing I would suggest is that the managers of collections already have a wealth of experience in how to create a biological reference library. So you need to make sure that we're included in those processes when you're thinking about how you're going to develop a library. And at that, I'll leave it there. Thank you.
Thank you, Jeff. That was really good, not least because of it showing the huge number of relationships that need to be captured when we're working in this space. And also it's interesting to see how Manaaki Whenua is actually applying the BC labels and the other labels to recognise Māori interest in the data. That's the last of the presentations. I'm now going to hand over to Nick to run the question and answer session. I'd like to personally thank all the speakers, since I probably won't get an opportunity to do that again. So thank you very much, and over to Nick.
Hello. Hi, Jeff. If you want to stop sharing your...
I'm just struggling with it at the moment.
Tēnā koutou katoa, ko te Manaaki Whenua, Head of Digital Strategy au, no Otatahi aho, ko Nick Spencer tako ingoa, tēnā tātou katoa. Well, thank you again to all our speakers and a wonderful series of presentations. For some reason my video's turned off, there we go. and a nice walk through some of the challenges with, as some said today, many questions that we're really asking without necessarily knowing the answers yet. So it's wonderful to be able to explore some of the aspects as we've done. And we saw some sort of specific New Zealand cases, some flavours of international, and some of the important components of any data system, but very particularly for this topic, things such as metadata, understanding the nature of the social component, and finally finishing with Jeff, where we heard how some of the significant biological collections in New Zealand are approaching this. So we've got a range of interesting questions. I'm going to do my best to read those out and then throw those to specific members of the panel when I think we've got the right association, but then otherwise I'll ask the general panel to please come forward with an answer. So starting with a question for perhaps Mick and Claire. Manpreet writes, I've really enjoyed your talk, learning a bit more about behind the scenes of AGDR. She's wondering if you have any feedback, if you'd like to discuss the nature of the feedback that you've had from iwi and hapu on their interactions with the data, and with issues around access approvals.
So the use of the labels is an easy one. So at the moment, we actually don't have any data sets in the repository that have got labels attached to them. What we've done is we've implemented the tools necessary to incorporate those, but we're then relying on researchers and their partners who are. Ooh, did we just start sharing our screen? Sorry, researchers and their partners having used those labels and included that information as part of their submission into the repository. Sorry, our AV is playing tricks on us at the moment. So we haven't got any feedback in terms of interactions, but we are looking at running some workshops with researchers and also with iwi research partners to talk about the repository and the use of labels. So we'll be doing that with the Local Context Hub team. And then the other part was around approval mechanisms. So at the moment, we've got one very heavily subscribed data set, which gets requested quite a bit, which is the Wānanga data set, which is contributed by PFR and is overseen by Rangatane. And so most of my interactions have been with them. And so there's been a very smooth approval process there because they have a very nice system in place. And other interactions have been tricky because we've had changes in Kaitiaki contexts occurring for data sets, and so I'm just keeping the conversation going with people to make sure we're talking with the right people when requests have been made. Have been, that takes time. The other thing is that researchers making access requests often don't provide enough information. So there's an initial screening phase to say, is there even enough information that's been provided to make a decision about access? When we go back to those researchers to get more information, often we don't hear anything back. So it's a very quick inquiry from them. We say, hey, we need a bit more info, and then we need to get back from them. So those are some of the experiences we've had.
Thank you. And I guess a follow-up question from Matthew, really. From Kieran, and he is asking a bit more about that question of how do you maintain contact with members of iwi hapu when there are changes, and the suggested model of email contacts might be a bit fragile. What are you sort of thinking towards improving that?
Yeah, and this is again very much an evolving process. So we're looking at what process can we put in place to maintain those connections more firmly, and make sure there's ongoing monitoring, ongoing engagement and conversations going on. Again, the difficulty is that those connections are made by the researcher. And so we're looking at how we can have a process where we're becoming involved in that relationship, we being the Genomic Data Project, becoming involved in the relationship as well, so that there's I guess almost a three-way relationship. That you've got your iwi, you've got iwi research partners, you've got the researchers themselves, and you've also got the data repository, and making sure that everyone's talking and understanding what the responsibilities are, and it's just ongoing communication about who is playing what role within each of those different organisations or entities. So watch the space.
Thank you. Now this might be a wider question for the panel, but Mick and Claire or Nimi, you might be best to answer this. So Bill has a question relating to the ability to store and access DNA data for pathogens, particularly for biosecurity purposes. Now, many New Zealand pathogens are often first discovered outside of New Zealand. So his question is, how will native New Zealand pathogen data be handled in DNA databases where there are concerns about international efforts to better understand these species?
Yes, no. So just very quickly, we're sort of kicking that around very quickly while people are talking, because we saw the question coming up. Again, this is one of those things that the conversations would need to be had with the appropriate groups, but you could imagine the ability, so within the system we've got, there's the ability to build tools on top of that. So you could easily query into the genomic data looking for a specific sequence or taxa without having true full access to all of the data. Now that would obviously have to be done in consultation with the kaitiaki or kaitiaki groups, but assuming that was favourable, you'd have the ability to perform those kind of, I guess, high-level interrogations of a data set without getting full access, and then if it became an interesting data set, you could then put it in an access proposal. And you do that in a user-controlled way where you may have to actually create a login to the system to be able to use it, so it's not just being bombarded, those sorts of things. But so it's, again, conversations happening, but that sort of approach would definitely be possible with the deployment we've got.
Cool, thank you. And this is a question for Lindley. Lindley Manpreet writes, thank you for the presentation. She's interested in hearing more about the balance of cost and benefit in terms of iwi hapu thinking, when we're partnering with scientists. What kind of benefits are you thinking do exist for iwi hapu in that context? And maybe contrast that with some of the costs that they would encounter.
Yeah, thank you. I think the benefits, plant and food is probably one of the more commercially oriented of the CRIs. And so a lot of the benefits that we've been working with iwi around is around the open to commercialization piece. So we've now started changing our business models with Māori in particular, that the IP can be shared, which is something that traditionally CRIs haven't done. And so we are working around sort of developing commercialization strategies with Māori and pushing that area, and that's in a range of different spaces. Costs are enormous. The costs of consultation, as Mick just mentioned, Rangatane is kaitiaki of the Manaaki genome. You should see the bombardment of requests that they get from this. So currently, we are resourcing Rangatane for a full FTE to deal with that. But that's short-term project-based funding. It's not sort of long-term relationship-based funding that is required. So I think this is a real challenge that we have, that CRIs tend to work at a project level. And so there's a beginning and an end of a project, but we're in our relationship with Māori, we really want that relationship level work where it's enduring and long term. So that's a big fundamental change in the way that we think about how we do science in Aotearoa. And so I think we have to have a big conversation about changing the model of science to enable these sort of things to happen.
Thank you. And I guess as an extension of that, you talked a little bit about the efficiency of working across CRI, and Robert asks, says, whilst that's great, but how do you imagine this broadening out to the wider Aotearoa, and to your point there of what the model needs to be able to support that in a sustainable way, perhaps? Absolutely, and I think with the green paper / white paper recently, I think there is an understanding now that research in an Aotearoa context, we really need to rethink how we're doing that. And so
I'm hopeful that we can start having those more national conversations, and maybe we should be coming together to put forward proposals, nationwide proposals to deal with research data, and bring in as many of the groups as different groups as possible to have these big conversations. I think that's a fantastic idea.
Thank you. Here's a question for Natalie. There are, of course, different knowledge bases held across different personas. Could you speak to any of the thoughts or ideas on how to create more regular exchange across the different actors in a data ecosystem, whether it be at institutional level or maybe more organic approaches across communities of practice, perhaps?
Thanks, Nick. That's a really great question. And we thought about this a lot. There were a lot of discussions among our team as we were developing these resources about this specific issue. And I think we need to see it as, well, from the biodiversity genomic researcher perspective, we need to have a bit of a shift of mindset to see our data management knowledge holders as our research partners, just like any other research partner that we may have. And that it's all about relationship building to break down these silos. So we can reach out early in the planning phases of research to get input on what's practical and feasible from our data management knowledge holders. A lot of these solutions are already out there. We don't need to be reinventing the wheel. And by reaching out early and often, we can be ensuring that our e-research teams are able to provide that wraparound support. So definitely it comes down to sharing regularly and often, being honest and genuine in those relationships, providing a lot of information up front so that everyone's on the same page, and not just being willing to share, but being willing to listen and learn and adapt as we go. And I think it's not just about going out and approaching our e-research teams, it's also about inviting them to come back to the table. So perhaps coming and speaking to the research group as a whole, inviting them to provide seminars and guidance and bits and pieces within our spaces as well. So I think there's two big challenges. I think there's a challenge to biodiversity genomics researchers to get out of the silo, go to where the e-research and library staff are. So maybe this is attending meetings or conferences that are more data-driven than research-focused. And I think there's also a challenge for e-research and library staff to promote the services that they offer, like seminars or drop-in sessions beyond teams or disciplines, and also to seek feedback on the implementation of some of the solutions that are provided. So it's a multi-level process, I think.
Thank you. And I guess picking up on your specific areas of focus around metadata, what do you think are some of the most important attributes of traditional metadata when we think about kaitiakitanga?
Ooh, that's a big question. I might actually throw this to one of our other folks, but definitely recording is key in terms of metadata. The sooner you record things, the sooner they're down on paper, then they're there and there's no chance of loss or having the data in the objects or the digital sequence of information getting divorced from one another.
Any other thoughts from around the panel? All right, then we'll move on. A question for Hamish, if you're there, Hamish. Bill is asking if you have any ideas why New Zealand isn't involved in the Centre for Plant Success, or maybe someone else in the panel or audience knows the answer to that question.
I'm actually not sure. I would speculate that it might be something to do with the ARC funding. So it's funded by the Australian Research Council, and they fund a number of these centres. And as far as I know, they're all based entirely within Australia. I don't think any of them have members from external countries, but I'm not completely sure on that.
Yes, that may be a reason, but perhaps there's an opportunity for some connections there. So Thank you. And Chris, I hope you're there. A question for you on the status of the eDNA module or services, and any lessons learned from running Geome through the Moya biocode project.
Okay, yeah, sorry, I'm coming in late here. Apologize. Well, you know, the Geome was set up initially to be specimen based, obviously. So, and that has been expanded to capture environmental samples. So it's become very event centric. So all the critical metadata associated with provenance and tracking is built into it now. And they're looking to expand that, particularly with the new GBIF ingestion models that are coming down the pipes, to make sure it can handle all the standards, metadata standards compliant with Darwin Core and Mix extensions as well. So we've dabbled with it for fish guts, for settlement plates, for filtered water and all that. It's not perfect, but it certainly tracks the critical things, particularly to provenance, like you're talking about. And it does have the capacity. I think John's presentation didn't show, but it does have modules to capture biocultural labels associated with the local context hub as well. So that's baked in as well currently. So that's all there. It's not perfect. You know, we're riding the bike while we're trying to, or we're fixing the bike while we're trying to ride it. But I think that's it's a good step forward, obviously, as you guys have mentioned.
And a very technical question from me. John mentioned that there's a good use of RESTful APIs, I suppose, in the architecture. Is there much uptake of that technology by people interacting with the repository?
It varies, I think. You know, I think it depends on how much people want to kind of use it as their primary, or kind of use it as a clearinghouse, you know, if that makes sense. So a lot of times we've used this as a cross-institutional clearinghouse where we use the information and then it kind of gets pushed to another system versus using it as primary. And then I think that more people use that more actively, if I'm understanding your question right. The other, I'm trying to think, I lost another thought there, but maybe I'll come back to it. Well, bear with me for a second.
No, that's cool. Thank you. Be interesting to interact with you on that offline, perhaps.
Mm-hmm.
Cool. And moving on, maybe a question for Jeff. Holden asks for any thoughts you have around how we might strike the balance between access to the data and affording Māori greater control over how that data is used. So over to you, Jeff.
Straight into the hard question. It's conversation. We're going to have a lot of talking ahead of us to think about how we're going to do this. I think in the short term, we're going to need to put gates in that allow how far we allow people to go in. For instance, at the moment, you know, any vulnerable species, threatened species stuff, we don't make that data available anyway. They have to come through it. So we'll put gates within the database so that people can only go so far and only see so much. While we have those conversations, I think, you know, Lindley made that point of there's going to be lots and lots of conversations. It's not going to be a fast process. I mean, I'm constantly talking about this being a generational piece of work, not something we're going to solve in the next 3 years or 5-year funding cycle. I think also with the examples we gave of 3 iwi looking at the material that we're holding, we need to expand that right out to the whole country. That one of the problems with our collections is that they're national collections, they're not just local collections. So therefore we have to engage with everybody everywhere all at once almost. And so that's going to be an interesting process in itself. And yeah, and often it's built around those one-on-one relationships. So we're going to have to figure out how do we manage those over time and space. And yeah. So I don't know if I answered the questions. There's a lot more problems out there than we realize. And well, I think we probably do realize it, but we would like them to be there. And I think there's also, again, with our funding models, we do have to slow down for some of this stuff. People are trying to do too much too fast, and we're not taking the public hapu iwi with us, because we're driven of this 3-year, 5-year cycle model. And as I say, it needs to be generational thinking.
And sort of extending that a bit, there are obviously even greater challenges when interacting or collaborating with international researchers and users. Any thoughts about how we extend that thinking, to that wider group?
Yeah. Well, can I say with Hamish's comments around Nagoya and stuff like that? I mean, we are taking that into account. We try to make sure we've collected all the permits, make sure that every specimen has some sort of permit or some sort of documentation sitting behind it that gave us approval to have it. As I said, the collections go back 100 years now, so we've got the bulk of the collection possibly had no permits or anything like that to start with, but then when they did have permits, nobody thought permits were important to keep. Permits allowed you to go in and get the stuff. Once you got it, you threw your permit away almost. And so finding permits that go back, say, before 1985 or something like that can be quite difficult. And also, issuing organizations change all the time. Who is responsible? For instance, my first permit came from the New Zealand Forest Service. Well, New Zealand Forest Service hasn't existed since '87. So, and I didn't keep the permit. So how do we find that permit again? And who has the right to give it? So, one of the interesting things that Manaaki Whenua is going through at the moment is its global permit from DOC to be able to go out and collect. But that global permit still requires us to consult with local hapu iwi, which is the way it should be. But from my perspective, we should already have that relationship, and we should be consulting with them before we go to DOC. And also, I think that there will be cases where the local hapu iwi say, we don't want you to consult with DOC. These things belong to us. So why are you going to them to ask permission to collect? So, there's a whole lot of social, political things that still need to be sorted out here.
And I guess you've partly answered one of the next questions that come to you, which was really about that challenge of historic data or collection when you don't have all the information. Any thoughts about how we work through that backlog? What are the appropriate changes to permissions and access that we should put around that data given the uncertainty?
Well, I think one of the things is the database that allows us to sort by rohe, we're going to be able to say, go to iwi, hapu iwi, and say, we actually have this, historically hold this collection from your space. Do you want to come and look at it? Let's start the partnership discussion about what we historically hold and work our way from there. It's going to take us a while probably to do that. As I say, there's not that many of us, and there's a lot of material, and even though we say New Zealand's a small country, when you've got to go out and talk to everybody everywhere all at once, it's kind of a challenge. But I think as we make it available, so that hapu iwi can come in and look at and see what we've got, that's going to enable it. And once we get a few of those done, then I think it will escalate and people will see the possibilities, and that will make it easier.
Thank you. And a question for Lindley that's been discussed in the chat back and forward. So maybe opening it up to Mick and Avery as well. The question was originally, what is the provision of resourcing and support for hapu to create our own data sovereignty management plans and systems? And maybe Lindley or Mick, you might want to offer a view, picking out of perhaps some of the discourse that's been going on in the chat.
Mick, do you want to go first? I can talk about plant and foods, but I think GA is probably more appropriate.
I tracked the comment in the chat there, and this is, I think it's an underappreciated issue, at least from our research side, that everyone does a lot of work, building these partnerships and maintaining relationships and engaging, but there's actually no funding for those being engaged with, and there's a huge amount of goodwill being used to maintain those relationships. There's a lot of time being spent in having discussions, particularly for the genomic data repository. If there's consideration that needs to be done for approving an access request, that takes time. There's no at the moment, there's no reimbursement for that time. There's no funding available for that. So that seems like a very obvious place that more funding needs to be obtained and allocated for, which is actually, I was starting to type a response in, which was basically saying full cost funding for research actually needs to cover the partners involved as well. It's not just the research costs. And I don't think that's ever really been addressed with thinking about curation of data.
Yeah, so from plant food side, we do resource a few iwi, not many, and we definitely engage with them with more than we resource. And those are at the project level. So the problem then becomes when the project finishes, the enduring work that still happens, like Manaaki Genome, for example, are still needed. So I think we do have to have that sort of more national conversation around a full costing of a research project, but what happens when the project finishes? How do we keep this going? And so resourcing Māori appropriately, I think, is one of the huge challenges that we absolutely need to address.
Yeah. So just to continue on that, there shouldn't be project-specific relationships. These are relationships between institutions and entities that should be very, very long-term. And so, the chat, I said we raised this in the paper that's been written about the AGDR. Basically saying, look, this needs to be centrally funded to allow these long-term relationships. So the responsibility needs to be taken by the institutions to create these long-term funded relationships.
Yep, absolutely agree.
Well, thank you. And it is a general challenge, isn't it, for research that's often project-based. And in the past, I think we've thought about this as very much how do we fund the ongoing, enduring infrastructure that supports data, but actually we're seeing there's a much more important aspect that's not often been considered up until recently around those relationships. So thank you for those comments. I'm going to go back to a thread of question we had earlier. This was concerning a DNA database and pathogens. And there was a follow-up response from one of the audience asking the question really about, given the the sense of contradiction really around the fact that international neighbours may share barcode information freely, yet we're unable to reciprocate unless one has to go through a controlled entry point. Mike is asking the speakers of their views of how to navigate this. Should barcodes be treated differently for genomes, given that they are used to listen to nature rather than bioprospecting?
Phil, are you there? So Phil Wilcox responded in the chat to Mike's combination thing. At least compared to me, Phil's far better placed to speak on this one.
If you're happy to speak, Phil, that'd be great.
Kia ora, Phil.
Kia ora. I guess it's in regard to Mike's question about the access to databases. It's stuff that we're still working through, and I see various levels of access are possible. Commentaries around, not sure if these things are known. I think that's changing. The environment internationally is changing, and that databases, there will be some level of, well, what appears to my mind is there's going to be in certain countries, there'll be some level of database limitation in terms of, or conditional access, I should say, not limitation, but conditional access. As I pointed out in the Variome investigation, the Variome project, we're working through that at the moment. Now that's a summary level database, and my comments were generally around that summary level database. I think Mike makes a good point around, well, if we're looking at having to look at access to the next layer down, which are the reads and such like that, contribute to that summary level database. But I don't see that as being problematic in terms of access. It's more about knowing whether or not these things are actually available, and that's about putting the message out there that conditional access will be available, as we have to do with the Variome. And when I say have to do it with the Variome, it's not have to do from the perception of the funding agencies. It's actually a moral obligation upon us in terms of what we have to do to deliver to our people.
Kia ora, Phil. And in the chat, Chris made a comment to this thread. Chris, I wondered whether you want to just sort of expand on your comments around data frictionless for identity.
I just commented that, we struggle with that same thing, because we want to create opportunities for, what we're calling, the blue or green economy here, develop tools around, sequence-based applications. But at the same time, you need to keep that connection to that data provenance for developing those kind of capacities. Whereas we don't want to, interfere with the ability to just flag something if it's important, especially early detection is critical for mitigation. So it's a really tricky needle to thread on that one.
Cool, thank you very much. I think we've come to the end of the questions that we have.
I do that every time I lift up the curtains, being like, light.
Someone needs to mute their.
I think there's one,
please. So with that, I'd like to thank all the panelists for responding.
Sorry. Sorry, Nick, we have one more question that's come through.
Oh, do we? Okay.
We should catch it while we can.
Do you want to read that out, Manpreet, since you've got it in front of you?
Sure. So Claire Adams is asking, how are we making sure we're talking to the right people, not excluding underrepresented or underfunded people? How do ECRs get involved, especially as science is increasingly diverse and ECRs aren't always in the academic space or may move between contracts rapidly? I think the question's around intergenerational relationship building. General question to the panel, I suppose, or
please, folks,
if somebody feels pressed. Maybe Lindy, you can speak since you're coordinating a group of pan-CRI representatives.
These are the big thorny questions, aren't they? Absolutely. I think the big change is that our research is getting more collaborative, and so there is a way now to bring ECRs into a process rather than go, there you go, ECR, go off and do your individual project and good luck and see you in 3 years' time. So bringing those big collaborative projects and bringing ECRs into those collaborative projects are really, really useful to make those, build those skills and then to change the way that we do science. I think how we talk to do science at universities is changing, but I think we need to continue that change around long-term enduring relationships, maintaining those. The work around the precariousness of ECRs, we all understand that, and that's a real challenge, and it's something I think we really need to address. I think that's a big problem that we have.
Okay, thank you, Lindley. Well, if that is the last question, but if we've missed any, rest assured we will find answers to any questions that we've missed, or maybe there's some additional aspects within the question that we haven't addressed. We will find answers for those and get those out to people. I believe the question and answer session will be also posted following this session today, as will be the recording of this Wānanga. So with that, I'd like to again thank the panelists for offering their thoughts and answers to an interesting set of questions, and to everyone who've been attending. I'll pass back to Manpreet to close us out. Thank you, Manpreet.
My job is easy in this Wānanga series. I just get to thank everyone for joining us today. And actually, if I could ask Sarah just to pop the slide on for the upcoming Wānanga. So once again, thank you all the speakers, thank you all the audience for asking all these important questions. I think this is exactly the kind of conversation we're hoping to ask questions and to give everybody an opportunity to think about these issues and to really assess, you know, the reality of doing this work is not very straightforward. We can see from the answers, we're all on this journey together. Everyone's trying their best to figure out a way that would work for our unique situation in Aotearoa. And with that, I would just like to highlight the upcoming topics. So the next one I'm sure will be of interest to many on data governance and sovereignty. And then following that, we'll be taking a deep dive into the technologies again. And the last one in September is on cross-project fertilization. We'll be looking at some other projects that have perhaps got interesting solutions for us to learn from. And with that, I would like to just say ka mihi everyone for coming today, and to invite Holden to close the meeting with a karakia.
Oh, kia ora, Manpreet, o tira, kia ora koutou katoa. Look, before I do close the hui, I just want to echo Nick and Manpreet's sentiments. Thank you all for joining. Very thoughtful conversation and kōrero. I think at the end of the day, for me, there's, you know, essentially what we're trying to do is build pathways whereby Māori can exercise greater control over Māori data. That's the long and short of it for me, and that's certainly what we'll be turning our minds to moving forward with these wānanga. So with that, I'll close our hui. Nā reira, kia taunga manaakitanga a te mea ngaro, ki runga ki tēnā o tātou. Kia mahea te hua mā kihi, kia toi te kupu, toi te mana, toi te aroha, toi te reo Māori, kia tūturu ka whakamaua, kia tīna.
Tīna.
Hui e tai.
Kia
Ora tātou. Thanks, everybody. Sorry, my camera wasn't working.
Thank you.
Ora.